Scientists may have found a way to more accurately track changes to our seas and oceans. They're using small samples of seawater and searching for the genetic clues left behind by passing animals. The technique is called eDNA, and Collin Closek from the Center for Ocean Solutions in California has been speaking to our science correspondent, Rebecca Morelle.
We're using techniques that have been used for the last 30 years or so for microorganisms, and instead we're using those same techniques for larger organisms. So we're using environmental DNA, which essentially is the skin cells or any DNA that is then suspended in the environment, and you can track that back to the organism that it was originally associated with. And so we're using that in the ocean water to try to assess differences in biodiversity that occur over time.
So essentially you're taking a little scoop of water and seeing which bits of DNA are in there, is that it?
Yeah, that's correct, yeah. And we're able to first assess the vertebrates that are in the water column, and then as we take water samples over time, we're able to see how that might change.
Why would you have to sort of do this rather than tracking the animals individually? Why use this kind of genetic approach?
So it's very resource intensive to go out and do assessments. Typically what's done is there's either fishing trawls that go through and assess the various species that exist in the water column, or from aerial surveys. But most of those are very limited in what they're able to capture. Many things go through nets, and they're also only doing these assessments at specific depths. And by looking at the water column, we're able to take a water sample, which is a little bit more mixed, and be able to assess how that changes or what diversity is associated with it.
I mean, what sorts of, just give me an example of a scoop of water and the sorts of things you've found living in there in the Monterey area.
So what I presented today was to assess the various forage species that exist in the Monterey Bay area. And so we were able to see differences from sardines to anchovies, for example, which oscillate quite regularly, and that's been known for the last century or so from different fisheries data. But we're able to assess that in the environmental DNA and then connect that with the various predators that might come in and eat those anchovies. So humpback whales or sea lions and various other mammals that come in to forage on those species.
I mean, how long do the skin cells or bits of fish or whatever sit around for? I mean, how do you know that your scoop of water is an accurate representation of what's there now?
So the residence time within oceanic water is on the scale of a couple days, whereas in other environments it tends to prolong a bit longer. And so we're able to use this in a way to be fairly certain that the organisms have been in that area for a very reduced time.
This is kind of the future for sort of keeping track of what's in our oceans. I mean, it does sound much easier than having to go out and see what's there.
Yes, we see that at least as being able to be a complementary method where we're able to collect more readily in smaller increments of time rather than on an annual or decadal basis, which a lot of these fisheries are assessed at. And so it would be nice to be able to collect them either via robots or be able to go out on some of these fishery expeditions and collect more samples and then be able to match that to some of the fisheries data that we're also already collecting.
That is Collin Closek of the Center for Ocean Solutions in California, speaking to our science correspondent, Rebecca Morelle.